Hi, I'm Trevor Moser, and I'm a scientist here at EMSL. In this short video, we're going to show you how to prepare samples and collect data for single particle imaging using our Krios G3I cryoEM here at EMSL. A purified protein complex is required for cryoEM data collection. Proteins can be sent already purified and ready for freezing, or we can use our custom cell-free expression pipeline to transcribe and translate the gene of interest in a test tube. A vector containing the gene of interest is mixed with wheat germ cell extract and incubated overnight. After expression, the protein of interest needs to be purified from the expression mix. For our pipeline, we use affinity-based molecular pull-down to separate our protein from the wheat germ extract. Expression and purity of the sample protein is then confirmed using gel electrophoresis. The sample can be concentrated or further purified using size exclusion chromatography. After purification, dialysis can be used to move the protein into a buffer compatible with cryoEM imaging. If your protein was shipped to EMSL in an already purified form, this step is where your protein would start in our pipeline. The protein concentration should then be quantified to ensure it is appropriate for freezing. For most projects, a concentration of 1 mg per milliliter in 100 microliters volume is appropriate. Next, the automated plunge freezing apparatus should be cooled down, the ethane condensed, and TEM grids prepared. Settings for blotting time and relative humidity should be set specific to the sample being prepared. The grids can then be loaded, sample transferred to the grid, and the sample frozen automatically based on the preset conditions. Frozen samples can be carefully transferred to a cryoEM grid storage box under liquid nitrogen. Care should be taken to avoid disturbing the nitrogen gas layer or exposing the grid to the atmosphere, as water vapor will quickly condense out of the air onto the cold sample and freeze as crystalline frost ice. For samples which will be loaded into the Krios, grids must be clipped to provide extra rigidity. Clipped grids are then loaded into the autoloader cassette and checked for proper orientation and successful clipping. The cassette is transferred to the microscope autoloader and docked. All samples are then loaded automatically into the microscope. Once in the microscope, data collection can be set up. Each TEM grid is then screened for ice and sample quality. In many cases, this is the most time-consuming step to find one grid preparation condition that is suitable for high-resolution data collection. Once a good grid is identified, suitable grid squares and holes are then selected for high-resolution imaging. Imaging conditions are chosen based on grid type and sample collection strategy, either for single or multi-shot imaging. Once ready, data collection can be started and can run unattended for days at a time. Once data collection is complete, image processing is performed. Typical processing steps for single particle analysis involves motion correction, fitting and correction of the contrast transfer function, particle picking and extraction, 2D classification of extracted particles, initial refinement, and final refinement. 3D electron density maps are generated and can be used for atomic modeling at resolutions beyond 2 angstrom for good samples. This was a brief overview of sample preparation and basic workflow for single particle imaging. Getting the most out of your particular sample, however, can depend on a multitude of different factors, which we'll discuss in more detail in the upcoming tutorial. Thanks for watching, and we'll see you soon.